Good afternoon. We want to present about the Plantibodies, antibody-based resistance against bacterial and fungal pathogens. Let me introduce our members. GRACELA NANDA with student number 169, SAFIRA TASYA with student number 171, AKHMAD RUDY with student number 172, SETYAWAN with student number 173, and CORYN ADELINA with student number 186. Next. Introduction backgrounds. Heterologous proteins production has increased efficiently. In plants, the number of heterologous proteins has increased due to advances in plant biotechnology. The demands of an expanding global population and an expectation of higher living standards are two main forces driving the widespread implementation of innovative agrobiotechnology concepts and techniques. Many crops have been genetically modified for high yield, stress tolerance, and production of diverse high-value products such as antibodies. Point 2. Heterologous proteins are plant-produced antibodies. Heterologous proteins, which often being discussed, are known as plant-produced antibodies. Point 3. Application of plantibodies have been explored. Due to its advanced development, application of antibodies have been explored since the first expert in tobacco on 1993. Example, an experiment on tobacco plant established its breeding and sexual crossing as a method for production of plantibody. In this experiment, transformation was used to introduce kappa type of light chain into tobacco plants. Next.
So in this opportunity, we are gonna discuss about four topics. The first topic is generating pathogen-specific antibody. The second one is antibody-based resistance against plant viruses. The third one is antibody, and the last one is antibody-based resistance against fungal pathogens. Next. So the first one is generating pathogen-specific antibody. So antibodies, also called immunoglobulins, are a group of complex glycoproteins produced by B lymphocytes and present in the serum and tissue fluids of generally vertebrates. They constitute the humoral arm of the adaptive immune system and specifically recognize and bind to target antigens on pathogens or toxin produced by such pathogens. When a pathogen enters a vertebrate host, antibodies that can bind to antigens on the foreign agents are rapidly elicited from B lymphocytes to combat and eliminate the pathogen. Antibody-based resistance is a novel strategy for generating transgenic plants resistant to pathogens. Decades ago, it was shown that polyclonal and monoclonal antibodies can neutralize viruses, bacteria, and selected fungi. This approach has been improved recently by the development of recombinant antibodies, or RABs. So the advantages of this approach are that RABs can be engineered against almost any target molecule, and it has been demonstrated that expression of functional pathogen-specific RABs in plants confers effective pathogen protection. Antibody which generate plant pathogen resistance relies on many aspects, which is target selection, careful antibody design, efficient antibody expression, stability, and targeting to appropriate cellular compartment. Next.
Okay, the next is antibody-based resistance against plant virus. There are 3 points. Number 1, the impact of virus-specific recombinant antibodies released on 3 factors. Quantity. There are yield and stability. Number 2, localization. There are this is intracellular targeting, and 3, binding efficiency. There are affinity and avidity. Point 2, Plant virus are the biotropic parasite, the type of virus that only is produced inside the plant cell environmental, and three structural protein and non-structural protein, which forms in infected plant. Structural point, component of virus particle that facilitate virus uptake. Nah, and non-structural protein needed for infection cycle but not part of the virion or infected from virus. Next.
Antibody-based resistance against bacterial pathogens. A study discussed on generating a single-chain fragment variable, or SCFV. It is a recombinant antibody and has great potential for use in many fields such as research. By generating this antibody, the major immunodominant membrane protein, or IMP, in store-bought phytoplasma can be recognized. Phytoplasma, known as a phloem-restricted plant pathogenic bacteria, so it is mainly restricted to the phloem sheath elements of infected hosts, or we can say that this bacteria is hard to deal with. But a previous study found a way to deal with this phytoplasma in a phloem using a single-chain fragment variable type 2A10 that was expressed in the cytosol of transgenic tobacco plants, which is active in the phloem among many other tissues. The antibody-based resistance against fungal pathogens. The investigation of fungal pathogens and their interaction with plants has established has identified several genes that confer resistance against pathogenic fungi. The varieties of genes resistant in the plant including enzyme and proteins. Enzyme for synthesizing antifungal metabolites, and two proteins. The first one is to inhibit fungal virulence, and the second one is inducing natural plant defenses.
From this presentation about antibodies, we can get the meaning of antibodies and antibody-based resistance. The first one is antibodies. Antibodies are a group of complex glycoproteins produced by B lymphocytes and present in the serum and tissue fluids of generally vertebrates. Then, antibody-based resistance is a novel strategy for generating transgenic plants resistance to pathogens. And then, about crop resistance can be engineered by the expression of pathogen-specific antibodies, antibody fragments, or antibody fusion proteins. And then, for the impact of virus-specific recombinant antibodies, was found to depend on three main factors like quantity, localization, and binding efficiency. That's all that we can explain about Plantibodies. For question and answer session will be done in comments column. Thank you for your attention.